Alright, this video is going to be on the effects of synthetic estrogen on fish, and presenting we have Ben Applegate, Mike Williams, and Roxanne Huff.
Estrogens found in birth controls are EDCs, which are chemical pollutants that can penetrate into exposed biota and alter their endocrine system by mimicking or counteracting the action of natural hormones. There are three main types of estrogens that have the potential to impact fish. E2, which is a natural estrogen, EE2, which is a synthetic estrogen, and nonylphenol, which is a weaker estrogen mimic. EE2 is the most potent of these estrogen mimics, and the majority of the impacts discussed involve EE2, but the impacts of the other estrogen mimics should not be ignored. Even in small quantities, in the lower nanograms per liter, which is the amount found in effluents from urban societies, chronic exposure can have profound impacts on fish reproduction, growth, and behavior. A study in 1999 found that 80% of river systems had pharmaceutical and personal care products in them, and it was not uncommon for multiple compounds to be found. The amount of pharmaceuticals, including birth control in the water, is expected to increase with population and is the highest near effluents in urban areas. Chemical pollutants enter the water through both point and non-point sources. Many synthetic chemicals and their derivatives are persistent or pseudo-persistent in their environment, meaning that they are slow to degrade and their continuous use leads to continuous release into the environment at a rate that exceeds the rate of their degradation. Human metabolism transforms EE2 into an inactive form, but degradation in sewage treatment plants and the environment reactivates them. The impacts of synthetic estrogens can be seen in both male and female fish, but are more evident in male fish. Exposure to synthetic estrogens has been shown to increase production of vitellogenin in both male and female fish. Vitellogenin is a protein that is typically produced by female fish. After only 7 weeks of exposure, vitellogenin levels were seen to be 3 orders of magnitude greater in a case study with fathead minnows. Males with higher vitellogenin produce early-stage eggs in their testes and may even become feminized. The effects on males severely decrease their reproductive success by impairing gonadal development, delaying spermatogenesis, malforming tubules, and decreasing sperm production and mobility, as well as decreasing courtship behaviors and decreasing the viability of fertilized eggs. Female fish exposed show delayed development of oocytes. The top 2 images show female ovaries. Figure A shows a healthy ovary with mature follicles, while Figure B shows undeveloped late vitellogenic cells. Figure C shows healthy testes, while Figure B shows early-stage eggs and testes.
In addition to reproductive complications, impacts from estrogens in the water introduced by birth controls also extends beyond the reproduction and into the fish larvae. Larval fish are often more sensitive to chemicals in the water than adult fish, and they show more adverse side effects than adults, just because they're a little bit more susceptible to the chemicals as they're growing. In fact, some studies have showed that the larval fish exposed to these estrogens have decreased their body size by 25% over those that were not exposed. So these chemicals interfere with development of both the central and peripheral nervous system, in addition to modification of the swimming behavior. And that changes can be seen in longer periods of inactivity and slower swimming speeds, which basically makes the larval fish more susceptible to predation. So those fish that are most likely to be impacted by those birth controls in the water play an important role in the ecosystem, and their absence or alteration has the potential to drastically change and affect other species that might not be as susceptible. And this can be caused by bioaccumulation and transference, basically, from species to species as they feed on prey that has also consumed the estrogen. So there is evidence that supports correlation between fish impacted by synthetic estrogens and larger changes in the ecosystem. So you can see at the diagram on the right, it displays roach biomass, which is a fish, and foraging success. with a control treatment and addition of estrogen to the water. And as you can see, the estrogen levels pretty dramatically affected the roach biomass and foraging success. So synthetic estrogen chemicals, as I just mentioned, accumulate in the river's trophic food webs. And these can be basically from trace chemicals from the environment and food. The problem with it is the removal and treatment of the estrogen chemicals can be elusive, and this is what allows for the accumulation in the river system. So accumulation is subject basically to the levels of human and animal use. A lot of it's used on farming, agricultural operations, and obviously for human hormonal control. The estrogen chemicals resist degradation in sewage treatment, and basically that equates to a mixture of stew of contaminants, and their accumulation is basically the huge concern here.
EE2's chemical composition is C20H24 and O2. Natural estrogen hormone E2 and the synthetic hormone EE2 have very similar chemical properties, the only difference being E2's carbon, which is only C18. The compound EE2 is nonpolar and hydrophobic, which makes it resistant to biodegradation. EE2 has been found to have a binding affinity to estrogen receptors of up to 5 times higher than normal. This abnormally high binding affinity means it is more likely to cause an estrogenic response, such as those seen in fish populations. EE2 has been found to be somewhat soluble in ethanol, with 1 part EE2 to 6 parts ethanol, but it is insoluble in water. Not only are synthetic hormones popular among humans, but they are also very popular among cattle farmers. EE2 is released in small amounts from cattle manure and urine. The hormone can then be absorbed into the soil and washed away and deposited into nearby water systems. It should be noted that hormones are also affecting birds and scavengers that eat the manure from the cattle. The runoff from agricultural land has little to no filtration, making the waters near these areas have especially high amounts of EE2. Currently, organic compounds are screened using persistence, bioaccumulation, and toxicity criteria. This is insufficient for determining pseudo-persistent chemicals or alterations to ecosystem functions due to cascading effects. Current toxicity testing requires a 50% mortality rate of test subjects to be considered toxic. This criteria ignores subtle ecological effects that can be just as harmful. EE2 is detected in very small amounts when it's passed through wastewater treatment facilities, and so it passes as acceptable. Many pharmaceuticals, like birth controls, are not classified as persistent chemicals despite having many of the properties of one. The lack of studying and regulations on pharmaceutical products and their impacts on water systems is caused by the perceived social benefit outweighing the environmental risks. Many compounds that resist biological breakdown persist long after their toxicity is discovered and their addition to water systems is halted. The rate of increase in the production and diversity of pharmaceuticals exceeds that of most previously recognized agents of global change. There is a gap in the impacts of synthetic chemicals and research conducted on their impacts. Less than 2% of research and funding is allocated to the study of synthetic compounds. And there are potential impacts for humans as well as aquatic life. For removal techniques, the first thing they go through is usually a removal of nitrates and phosphates from aquaculture filtration systems. And then there's the use of polyimide particles, and they surpass the removal capabilities of activated carbon in their benchmark removal techniques. But the most effective one that has been found has been activated sludge. So the image, sourced from Aris et al., shows tested removal techniques and their accuracy. Nitrifying bacteria in activated sludge was found to be the most successful at the degradation of EE2. The process of nitrification is able to degrade EE2 in the absence of ammonium. Nitrifying bacteria has capabilities to react on low molecular weight compounds, making it an excellent addition to wastewater treatment plants in order to break down synthetic hormones before effluent is released back into water systems. Ozonation is also a common technique used by wastewater treatment plants, but studies have shown that the process can cause EE2 to become more harmful. Ozonation is a process in which O3 is infused into the water. EE2 showed little to no effect on estrogen levels, but showed a negative effect on testosterone secretion. So the impact of EE2 with O3 may have a more toxic byproduct than the parent material. And then on this last slide, I just wanted to add this in here to reiterate how well nitrifying sludge is at the breakdown of EE2. The image is from a study which focused on nitrifying sludge use on EE2. And this graph here shows the degradation rate of EE2 using that nitrifying sludge. The sludge was able to completely degrade EE2 within 6 days. Although this experiment was not conducted at a wastewater treatment plant, it shows promise that we can add solutions such as this to our treatment plants to help reduce EE2 being released back into the environment. And lastly is our reference page.